if you don't mind. So as you see that the title of my talk is very long. Small and X-ray spectrum studies of the polymerase state and the quaternary structure of the trifunctional proline utilization chain, or in short we call it PATE, which is a flavoprotein from E. coli. Before I talk, let me give you a brief outline of my talk. First, I will start with a brief introduction of my talk. Then I will talk about the research goal in my project, followed by the current results, and at the end I will talk about some conclusions. So my lab extensively worked on proline catabolic enzymes. In proline catabolic, proline oxidizes to the final product glutamate, which is actually a 4-electron oxidation process. So in the first step, proline oxidizes to an intermediate pyroline 5-carboxylate, which is catalyzed by an enzyme, which is a flavoprotein, proline dehydrogenase. And then after this pyroline 5-carboxylate, or we call P5C, non-enzymatically hydrolyzed to glutamate semialdehyde. And at the final step, the NAD-dependent pyroline 5-carboxylate dehydrogenase oxidizes into the final product glutamate. So overall, this is a 4-electron oxidation process carried out catalyzed by these two enzymes. Throughout my talk, I will be calling proline dehydrogenase as ProDH and delta or pyroline 5-carboxylate dehydrogenase as P5CDH. So in many organisms, these two enzymes work independently, so they are monofunctional enzymes. But let me confine myself to one of the special kind of organism where these two enzymes are actually fused together and they work. They are coded by the same gene, and they are from the gram-negative bacteria. They are in the they exist in the gram-negative bacteria. So they are actually a bifunctional protein. And in short, we actually call it proline utilization A, or I will be calling throughout my talk PATE A. So one of the examples of bifunctional PATE which our lab extensively worked on is PATE from Bradyrhizobium japonicum PATE. Throughout my talk, I will be using this enzyme as BJ PATE. There is another class of PATE which has DNA binding domain at the N-terminal region of the protein. So it's a autotranscriptional repressor also. So along with these two, the two functional activities for proline catabolism, it also have a transcriptional repression activity. So we call it a trifunctional PATE. One of the examples is the PATE from E. coli. In short, I will be calling it EC PATE or E. coli PATE and so on. So before let me give you in this slide, let me give you the key differences between the bifunctional and the trifunctional PATE. So this diagram is showing you the color-coded sequence alignment for the bifunctional as well as the trifunctional PATE. So the top one is the sequence of a bifunctional protein represented by a 999 amino acid residue long BJ PATE, and the bottom one is the sequence of a trifunctional PATE represented by a 1320 amino acid residue long E. coli PATE. So as you can see from the color-coded sequence alignment, most of the features, most of the domains and the motifs representing which are responsible for catalyzing the enzymes are common in both of the enzymes. The key difference we see here is that there is at the N-terminal region there is a DNA binding domain, and there is the C-terminal domain. There is an insertion between the NAD binding domain and the predicted C-terminal motif. So this N-terminal region makes it an autotranscriptional repressor, and it can switch its function from DNA binding activity to proline catabolic activity when it binds to the membrane. So actually it switches its location. This trifunctional product switches its location from DNA binding to the membrane binding protein. So we call this phenomena a functional switching, which is a part of my project to work on. So as far as this bifunctional protein is concerned, we have the overall crystal structure of the protein from our lab published earlier, where the C-terminal domain is involved in the polymerization of the protein. So there is a C-terminal motif called beta flash, which is involved in the dimerization, and actually it's a dimer of a dimer that makes it a tetramer. What is known about the overall trifunctional protein is the crystal structure of the isolated DNA binding domain, which comes into the ribbon helix family. That the crystal structure is it's a ribbon helix. It shows that it comes in the ribbon helix family. It's a diamond, and also we have the structure of this protease domain, which shows that there is a alpha beta barrel, which is also present in which is also common in the bifunctional protein. But the overall structure of the protein, or the C-terminal structure and overall structure of the protein, trifunctional protein, was always elusive. So, of course, the first thing to do is to try to attempt the crystallization of the overall trifunctional protein, full-length protein, check the oligomeric state of the protein, and of course, the crystallization step was a sad story for me. So it's later on do the same reconstruction using the small angle X-ray scattering, do rigid body modeling, and understand the functional switching phenomena from DNA binding activity to the membrane binding activity. So let me start with my sad story. So here is the E. coli, the crystal of needle-shaped crystal of the E. coli protein, which I got under oil. It's a needle-shaped crystal. Doesn't look. It's a flavoprotein. Should look yellow, but it's not. Doesn't look that yellow. I got some diffraction. Kind of poor diffraction at advanced photon source. It's around 7.6 to 6 angstrom. Detector distance is 1000 millimeters. Still the spots are very close together. Unit cell dimension, as you can see, kind of discouraging to work on the project in the protein molecular weight is around 290 kilodalton. I moved to the one homologue of the trifunctional protein, which is from Salmonella typhimurium, 90% identical. Just a usual protocol in crystallization that maybe it can diffract better. So from the needle-shaped crystal, actually I was able to optimize it to get it this Salmonella typhimurium patent to a dark-looking yellow thick crystal. So it was very encouraging to work on it and spend some time on it. But all to get the same kind of diffraction, poor diffraction with the same detector distance, still not working. So, as a graduate student, I have to do something to finish my PhD. So the part of the blame always goes to the size exclusion chromatogram of the purified protein. So, as you can see that there are two overlapping species, and as you can see, the first one is the dominant, and the second one is always hard to. If it's a soldier peak, it's always hard to isolate the soldier peak. So we have isolated the first peak, which is actually 20 years ago proved by Janet Woods as the functional dynamic protein, and that's what I should work on because that's already been worked. So I just pulled this fraction. At least it looks good on the size exclusion chromatogram. I don't know, I am able to separate that or not, but at least it looks good on the size exclusion chromatogram. So just doing multi-angle light scattering to know the first peak, which was proved by our earlier scientists who used to work Janet Wood, that it was a diamond. I proved through multi-angle light scattering that the protein, this blue curve is actually the molecular mass of the protein, and this is the response from the refractive index detector, and the black curve is actually refractive index detector response, and the red curve is light scattering response at 90 degrees. So this curve is not actually perfect straight line, it all depends on the scale, but actually it's homogeneous, and the molecular weight is around 6% of the 290 kilodalton, which is the diamond molecular weight of the protein. So protein is a diamond. I just checked it, verified it. The next thing was to do so right now I don't know which part of the protein is involved in the oligomeric state. As I told you that the bifunctional protein has a conventional C-terminal oligomerization flap which is present in all kind of aldehyde dehydrogenase. Maybe I didn't say that it comes also in the aldehyde dehydrogenase family, which has a conventional C-terminal oligomerization flap called beta flap. So we didn't know that this kind of what oligomerization domain it has. So as I already showed you that N-terminal was actually N-terminal is a DNA binding domain, and it's a dimer. So it makes us think that maybe the N-terminal is dimerization domain. So let's first look the small angle X-ray scattering experiment. So these are the scattering curves for the 5 different samples in different concentration for the full length E. coli protein. So the y-axis is intensity as a function of the scattering vector represented by this formula. So These are the five scattering curves from the different concentrations. Maybe it is not quite obvious to see the depression and the bump at the arrow shown here. Can you just see the depression and bump? Maybe not. Anyway, so I can just make it clear later on. So there is a characteristic depression and bump at around 0.05, 0.04 and at around 0.06 per angstrom of the protein, which I think is the characteristic of my trifunctional protein. The figure in the inset is a Gaussian plot, which is a perfect straight line in the region of QRG less than 1.3, telling me that protein doesn't have any aggregation or particle interference problem. And I have done a lot of multiple samples, so it kind of makes sure that there is no problem in the protein. So as I showed the signature of this depression and the bump, which can so this scattering profile can be transformed into the pair distribution function, which is actually the histogram of the interatomic distance between the protein within one molecule. So and is the signature of the shape of the protein. So as you can see from the 5 samples, all of them had a major peak at around 43 angstrom and a soldier peak at around 103 angstrom, telling me that the protein is actually most of the electrons are situated at the random distance at the distance of around 43 angstrom, and around some of them are separated at around 103 angstrom. So this pair distribution function is more like the Patterson function of crystallography. The only thing, the difference is like it just says the interatomic vector distance within the molecule, and I don't know anything more than that about Patterson's function. But anyway, let's see the shape reconstruction of the protein. So I use the dashboard shape reconstruction. So all of the 5 samples, 10 independent jobs from each of the 5 samples under investigation. So total 50 jobs, and did the shape reconstruction using the software dashboard. So as we saw that in the pair distribution function, there are 2 peaks. This shape reconstruction is actually consistent with our pair distribution function. So it has the 2 lobes connected by the small connector region. Maximum dimension is 205. I didn't talk about radius of gyration. Radius of the gyration is around say 62 or 63 angstrom. So this is how the shape of the protein looks like. So now the question is, what is the protein looks like a 2-fold symmetric dimer. So the question is, what is the dimeric domain? Like which part of the protein is involved in dimerization? So as I already told you that the reverse helix N-terminal DNA binding domain is dimer, and it binds to the DNA as a dimer. But I cannot take it as a granted. So we made following constructs. So the first one is Patek 86 to 1320, which doesn't have DNA binding domain. So if a DNA binding domain is involved in dimerization, this truncation should be a monomer. And the second truncation is Patek 1 to 1085, where I deleted this part and truncated up to here, and this kept the DNA binding domain. So that should be a dimer if the DNA binding domain is involved in dimerization. So the multi-halide scattering for Patek 86 to 1320 says that the molecular weight is around 140 kilodalton, 144, which is around less than 5% of 135 kilodalton, the molecular weight of a monomer. So that tells me that the DNA binding, truncating the DNA binding domain actually has affected the protein so that it became the monomer. So here are the scattering curves for the comparison of the scattering curves of the domain deletion mutants with the wild-type protein. So the first one is the E. coli Patek, full length. The second one is Patek 1 to 1085. As you can see, maybe you cannot here, but the depression and the bump are consistent in also in Patek 1 to 1085. But once you go to the Patek 86 to 1320, that completely eliminates that depression and bump characteristic. If you are not clear here, you can see it in the pair distribution curve that the Patek, E. coli Patek and the Patek 1 to 1085 have similar kind of pair distribution curve. But once you go to Patek 86 to 1320, you completely lose the second soldier peak, and also the radius of gyration peak is from 60 angstrom telling me that my protein has the DNA binding domain involved in memorization. So these are some of the parameters derived from the small angular X-ray scattering. Maybe I don't have the time to right now talk about it. But anyway, I already talked about what it is. So the next thing is to know the spatial arrangement of the protein dehydrogenase, P5CDH, and the DNA binding domain. For that, I did some rigid body modeling and put the two-fold axis of the crystal structure of DNA binding domain at the coincident with the two-fold axis of the volumetric map of the protein. And then I model pertain the residue 86 to 1130, which is the hybrid of homology and homology model of X-ray and X-ray crystal structure, and homology model of 86 to 1113. I did not put the C-terminal domain at this point, but it is the protein, the model is almost complete. When I reverse calculated the scattering profile and compared it with the experimental profile, I saw the chi value, the fit of the experimental with the model is quite well. Keeping in mind that we still are missing few 200 residues. Still we are getting a fit like chi square value at around 2.7, which is much better, and also the characteristic difference and beams are bumps are quite characteristic here also. Next thing was to do the DNA binding. Next thing was to just look for the model of the DNA bound protein. This picture is just to show you that how DNA. We also have the DNA bound structure. So I did the same thing. Coincide the two-fold axis of the DNA crystal structure of DNA binding domain to the volumetric two-fold of the volumetric map, and you can see that this DNA binding may be affected by the N-terminal, the protease domain, the alpha arm here. It can be affected, which is consistent with our results that the DNA binding affinity is quite low compared to the isolated DNA binding domain with the full-length protein. So it gives us also some idea. This model also gives us some idea about how it interacts with the membrane, because we know that the reduced protein binds to the membrane higher than several order of magnitude than the oxidized protein. Though the DNA binding domain doesn't affect. It's just telling me that the DNA binding domain, there is some conformational change after the reduction of the protein, which doesn't affect the DNA binding domain, DNA binding activity, but it affects the membrane binding. So what I did is I used this reducing agent. This is a covalent reducer, like it's covalently bound to the NAD and reduces permanently. This is the substrate of the protein, and this is the inhibitor. So I can see these are the multiple pair distribution curve for the full length E. coli pate reduced with the covalent reducer, irreversible reducer with the substrate, and as well as the inhibitor. So I can see there is some changes in the. As I already told you that the pair distribution curve is significant of the shape of the protein. So there is some consistent change in the second peak. It leads us to idea that there is some conformational change, there is some twist in the structure. Right now I don't know, this is just the initial result, but it gives me some idea that the radius of gyration is quite increased here, consistently around 65 angstrom, then 61 or 62 angstrom. It is giving me idea that maybe I should proceed this and do a time without small angular X-ray scattering and see how it behaves there. So the conclusion is we know that the oligomeric state of pate are now not conserved. One of them bifunctional was a tetramer, though the one which we just see is a dimer. The first three-dimensional structure of information for any trifunctional pate is just being reported, and the spatial separation of the P5CDH domain, which makes it aldehyde, comes into the aldehyde dehydrogenase family, is separated here. Though in the aldehyde dehydrogenase family, that's the one which makes the oligomerization domain. So this thing is unprecedented and exceptional. So it also gives the insight of the global conformation change while the functional switching the protein. So at last I want to acknowledge my boss who has let me work in his lab, though I don't think I deserve that much. So these guys, I don't know how to drive, so they just drive me here. Posters and my friends and graduate students in the same lab. Kevin Dyer, who has collected the online beamline checks, all my checks data, and also the organizers of Mid-Atlantic macromolecular crystallography meeting. to let me talk, have my first talk here. Thank you very much.
For the DNA-bound form of Part A, I was just a little bit confused. Did you actually do stacks on the DNA-bound form to get the envelope, or did you, or was that the envelope of DNA-free and you ended up purely a computational document of DNA?
We have the crystal structure of DNA-bound, DNA binding domain, and I docked it manually as a two-fold axis of the volumetric map. And I just assumed here that it goes in the connector region, the small cylindrical region, because there is no other space to go. So it's an assumption.
So no stacks on the complex?
No. The binding affinity was quite bad. Like I tried it, but it didn't work.
So you showed us the stacks data. You showed us the stacks traces for the truncation unit that doesn't have the DNA binding domain, but you didn't show us the envelope.
That's a very good question. Because actually you cannot do shape reconstruction for almost anything. Like there is some flexibility. The protein is very flexible. Yeah, once I remove the DNA binding domain, somehow it becomes like some part of the domain becomes very flexible, and when you make it, when you do the shape reconstruction, it looks okay, like it looks like half of the protein, but there is a part of the thing which is very extended. So the shape, ab initio shape reconstruction is not always reliable according to me to just make room for almost anything. So I just didn't do it. I did it, but I don't show it.